I appreciate this opportunity to address this audience for whom I have a great deal of sympathy. Remember, I was involved in Max Moore's original Extropy conferences that were held here in Santa Monica back in maybe 10 years ago. And I've been attending some of these meetings whenever I could, like there was one in Irvine not long ago. And I thought we were going to have this one not here at Caltech, but at UCLA, my home venue. But it turned out that this was a little bit more convenient. And not only that, I have a sentimental attachment to this room in particular because I've attended lots of lectures here by Nobel Prize winners, including the former president of Caltech, David Baltimore, and as well my old mentor, the late Joshua Lederberg, who was a Nobel Prize winner in medicine as well from Stanford University. And I remember he said a famous quote. He said, "Aging is programmed." And if you look at all the mammalian species that I'm going to show you in one of the slides, there are mammals that live only a year. And then there are others that live 2 or 3 years, like mice, and rats live 4 or 5, and cats and dogs, and then all the way up to elephants and humans, and bowhead whales who live 225 years. And we don't understand any of their secrets except that somehow it's in their genome. And I started looking at this problem in human beings, maybe 15 years ago, and I interviewed lots of people who were very, very old, asking them, what's your secret? Is there a limit to your life expectancy? And they would usually give a comical answer because lots of their relatives and family members asked them the same question, as well as reporters who want to know. And they gave contradictory answers. It's because I drank Jim Beam whiskey every day, or because I've never drunk anything at all, or, and some people, like George Burns, who lived to 100, it's because I smoked 2 or 3 cigars every day. And we know that's not right. But yet a lot of the supercentenarians I interviewed smoked because that was fashionable back in their early years. My parents smoked. Everybody amongst my parents smoked as well. And I don't know, what it is, but when I started doing a careful history taking of these people and their family history, I discovered that they did have one thing in common, which was their first-degree relatives. Their parents, their siblings, probably their children were all long-lived. So undoubtedly, it was in their genes, in the DNA. And if we could only get that DNA before they passed away, because it's a very fragile community, the oldest old, then maybe we could do a complete sequence and compare them with ordinary people and see what was unique and special about their inheritance. Well, the quick answer to the question at the top here is there is an upper limit, but there is a footnote on that answer, which is, well, all bets are off if we can do something about it. And so I am especially focused on interventions once we discover the things that they have in common genomically. To show you that my heart is in the right place, I'd like to quote Alan Harrington back in 1967, in which he said, "Death is an imposition on the human race and can no longer be tolerated." And I said, oh yeah, that sounds interesting. Where do I sign up? So I found where he was. He was living in Manhattan at the time. I went to visit him, and it turned out he was working on his next book and not very interested in discussing the topic much further. But there were famous gerontologists like Alex Comfort who wrote the foreword for his book. And at the time, that was a very influential book in my thinking. I want to also explain the difference between biogerontology, which is what I do, and geriatrics, which concerns the diseases of old people. And I mean, I have spent time in nursing homes, and I understand a thing or two about geriatrics, but it's essentially internal medicine for people who have age-related diseases. And biogerontologists are more generalists. They look at yeast and microscopic worms like C. elegans, flies, primates going on the way to humans. And in my personal history, I looked in 1960 at a claim that was made in Playboy magazine in an interview with Sir Arthur Clarke, in which he said, we ought to have an institute for studying immortality. So I wrote him a letter, and he actually wrote back to me and said, I was just kidding, but if you want to do it, good luck. The A4M, an organization especially devoted to anti-aging medicine, started its first meeting in 1993 in Mexico, and then for the next 10 years or so, in which I was a speaker in that group, a couple of times at least, they met in Las Vegas. And then when I had a split with the founders of that organization, I'm sort of a persona non grata now for those people, we started a parallel set of meetings called AMMG, the Age Management Medical Group, and we've met both on the East and West Coasts here ever since. Aging is sort of like the blind men touching the elephant, because it has a very different shape if you're blind and you don't know where you're touching, okay? Here there are 6 blind men according to the, tradition in India, and we have so many different issues to focus on in gerontology that everybody has become a specialist, and they tend to have different theories, like it's in the germline, or no, it's in the protein misfolding, or it's in oxidative stress, or mitochondria, or neurological problems like the clock of the circadian rhythm in your brain, endocrine theories, immunological theories. Inflammation, of course, is important. Stem cell depletion is another issue, and maybe the accumulation of garbage or trash that we don't know how to get rid of in our cells is a key. Well, I think up here somewhere I mentioned telomeres, and I should say that just last Sunday in the online journal Nature, they described an experiment in which it looked like telomeres were the answer until you read the fine print and not just the headlines in the newspapers. In fact, I have the original paper, and it's got a dozen co-authors on it, and Ron DePinho, the last co-author, is a very senior member of the academic gerontology establishment. I mean, he's an MD who has worked on cancer for 30 years, and furthermore, he's a member of the National Academy of Sciences and the Institute of Medicine, and here he is saying to the press, we have the secret, and it's in telomere length, because our mice, just looked very old, and when we fed them telomerase, which is the enzyme that re-lengthens telomeres, they look great. They looked young again. We not only stopped aging, we reversed aging. Well, it turned out if you read the fine print, they broke the machine before they started because they did a knockout of that telomerase gene. It's almost like building a car without wheels and saying, gee, this car doesn't work. And then coming back 6 months later and putting wheels on the car, and oh, it's a miracle. The car now drives, And in that experiment, the mice didn't live any longer than control mice that were never played with in the first place. We're in the dark ages of anti-aging research. I claim that we're in an alchemy stage where lots of people are doing lots of things. And here is the real wizard reading the real book, okay? And getting advice from somebody looking over his shoulder. But somehow the science has to move along to the stage of the periodic table of the elements in which we really understand how things are put together. And we can make sense out of the recent discovery that there's a form of bacteria in Mono Lake here in California which uses arsenic instead of phosphorus. And it turns out that if you look over here in the periodic table, it makes sense. They're next-door neighbors. In fact, it explains to a large extent why arsenic is a poison, even though it's not a poison for those particular bacteria that use it efficiently. The Future of Aging is a book that came out on the market from Springer. Here's a copy of it. It's like 750 pages talking about our latest theory. And I co-edited this with Michael West and Greg Fay and Stephen Harris, who is my personal physician. And there are lots of things that are not covered in the book, like heterochronic parabiasis. When you combine the bloodstream of mice that are both young and old as a common supply, it turns out that the old mice get a little younger, the young mice get a lot older. And so we know there are proteins circulating in the bloodstream that are very important for the stem cell population for growth in old age. There's a grandparenting effect. This interesting diagram here shows in C. elegans, in microscopic worms, a huge right shift in the longevity curve because of a change in a single gene called age1. Queen bee and queen ants are fascinating insects. They live 10 to 15 or even 20 times as long as the worker counterparts. And they have the same genome. There must be something special about their epigenetics, because queen bees to be are eating ambrosia, a special food that the workers don't get to eat. That must have some implications. Of course, people thought of that 20 years ago. You could buy on the internet ambrosia for humans, but it obviously didn't do much for us. Throughout all of history, there have been very few humans on the planet, looking at the area under the curve from 2.5 million years ago. But suddenly, in the last century, things have taken off, and there's been an explosive growth of humans on the planet. And our average life expectancy appears to be rising exponentially. That told me something about the centenarian population when I saw that was also rising exponentially, that must mean that the Calmet limit, the woman who was the oldest person in history in the Guinness Book of Records, would have her record broken very soon, because it's a real horse race. Well, turns out that the Bible had some opinions about this. The antediluvian patriarchs, the people who lived before Noah and the flood, lived a heck of a long time, including Methuselah, 969 years. And it turns out that we actually have people who reflect our table E of the oldest people living in the world today in a Wikipedia format that they criticize because it contradicts the Bible. There are myths, of course, the Fountain of Youth, Ponce de Leon in St. Augustine, Florida. And there are, I think, that electrical connection may have. I think it's plugged in. Let's see. This is on, so that's okay. Yeah, I apparently have been running on a battery without being recharged.
I think I just wasn't stuck in them.
It's possible. When computers get moved, they're very unhappy. Humans likewise.
It's going to reboot now?
It's going to resume, hopefully, quickly. Well, I picked up where I left off. Very good. Now, the wizardry of the Microsoft operating system. Let me go through, this slide you've already seen in Greg's talk before me. This is the exponential increase in the centenarian population, but I don't really study centenarians because they're youngsters by the standards of super centenarians, the people who are 110 or older. And we want to know Why do they live as long as they do? How do they escape from all the conventional diseases that take out the ordinary population of heart disease and cancer and stroke and diabetes and Alzheimer's disease? And if they are escapers, then but they still die. Why don't they live longer still? What are their causes of death? The only way to know that for sure is to do autopsies. And then what can we learn about them? By studying them so that we can have a better feeling for the underlying causes of aging that impact everyone. And I'm going to go quickly over some statistics to fit the talk into the time, but there's a couple of things you should know. There are only 82 supercentenarians living in the world today, and they're sort of randomly distributed geographically. And of the ratio of females to males, it's extraordinary in a female advantage. You women in the audience have something special in your genome that makes a huge difference at the very end of life. Where is your stamina coming from? I'm not sure. But it could be more than 10 to 1 difference. Some people theorize that it's due to estrogen, but I don't know whether that's true or not, because it could actually be the double X chromosome, because the men's Y chromosome doesn't have very much in the way of genes that are important, other than growing hair on your ears. Okay. Here is a picture of what Jeanne Calment looked like when she was 40. Here she is at 120. She was very healthy despite the fact that she was smoking at 115. And yet, when we look at the Japanese supercentenarians, the oldest person was 114, even though there were 3,000 centenarians. And there were 40,000 in 2009, and still the oldest person was only 114. This is the famous Gompertz curve of mortality as a function of age. And when you're born, there's a lot of childhood diseases that can afflict you that will, if you manage to survive to age 1 or 2, cause your mortality to fall to the minimum that you will ever have for the rest of your life, which is at puberty. Then it rises again, it sort of plateaus between 20 and 30, and then it rises in a straight line fashion, which is actually exponential because it's a semi-log plot. And what happens out here is still a mystery because there are too few data points to look at. I'm not going to go through all of these details other than to mention that in different countries other than France, where we had this outlier, Jeanne Calment, the oldest was 115 or 114 or 114, and other European countries where record keeping was done with great accuracy in the year 1900. Now, one of the things that Michael Rose would love to talk about in his experiments with fruit flies is this plateau phenomenon, which appears in the oldest old of the fruit flies. And there is a lot of controversy about whether that kind of model in which you can have a sort of linear plot for the first deaths and then another model of what goes on in the oldest old fruit flies, whether that applies to humans is not known. Let's see. Yeah, there's a whole lot of data that we've accumulated over the last 15 years, and when you get out to the fringes, when there are too few data points to plot, things look a blur. And I'm just going to mention some mortality statistics that we have uncovered at 49.9% for people who are reaching 110. The probability of living even one more year to 111 then is like flipping a coin. It's very scary that you won't make it to the next birthday. And then the mortality rates actually go up. 48%, 53%, 55%, 67%, 68%. Depending on how you plot it, in half years instead of years, it can go up to 70%. So mortality rates are rising even in the supercentenarian population. This is the latest data as of November 27th, and it's rising right around 42, 43%. And then there are too few people, like 4, 2, or 1 person left to even make an assessment of these statistics. The conclusion here is that the mortality plateau, even if it exists, is not the end of aging, because no one of our supercentenarians has ever had an age reversal or an aging plateau of the phenotypes of aging. They always get more frail, more deaf, more blind, and sicker in every way. But our hypothesis is that the data of the 22,000 genes must be very important. We've got to find out what that is. And I'm going to skip over some slides. This is the one, by the way, about mammals, and the range, the dynamic range of the phenotype of life expectancy or maximum lifespan is enormous until you hit these magical bowhead whales. We haven't done the DNA sequence of whales yet. We've been trying for 10 years to get the attention of the people who do the sequencing, but it's a hard problem. Here are the leading causes of death. We're all aware of that. Here's a repeat of one of the earlier slides. Here's more about the female advantage. It looks like the female advantage is increasing as a function of age, even past 110. And here's the records on what we call Table D of the oldest people in history from, I guess, back in 1745, when it was 111. And it went up to 122 with Jean Calment. And there's a question mark after Izumi's name. This is a gentleman from Japan who was alleged to be 120 when he died, and now we know better because of a necronym in which the parents named him for his brother who was 15 years older when he died young. And so as a result, really Izumi was only 105. So he is taken out of all the new statistics. And I have lots of data and graphs to explain that there is a plateau in the numbers of supercentenarians starting in about 2005. Here's the list of autopsies that I have participated in at UCLA and 2 or 3 other medical centers. And TTR is the diagnosis in 1, 2, 3, 4, 5, 6, 7 out of the 9. What's that? I mean, I could give another 1-hour lecture about transthyretin as a cause of death in these older people, because it is a disease process, a pathology that is occurring in all of us and rises exponentially with age. And the reason why it shows up in these people is that they just lived long enough to manifest this other form of the Grim Reaper if you escape from the heart disease and cancer and stroke. We did the autopsy of Milan Herzog, who was a famous film director in Hollywood here back in April. And here he is with his wife, Shanta, and his final autopsy report says what I was saying, that he had amyloidosis of the heart, of the TTR form. This is Dr. Fishbein, who is looking at the heart and holding it to see what the amyloid looks like on gross inspection. Of course, you don't know what form of amyloid it is until you do additional analysis with staining, like Congo red stain and trichrome stain, to see what it really is. But we knew that he had some form of dementia, and we asked Harry Vinters, who is the neuropathologist, if he could look at the brain in detail. And when we looked grossly at a brain cutting, there was nothing obvious. It was not at all conspicuous what was the cause of dementia by slicing the brain and looking for evidence of a stroke, because there wasn't any. Alzheimer in the early years, and we discovered that in this is a physician from Germany, a man and wife team, Brock and Brock, who developed a scale on Roman numbers from 0 to 6, and our case was a Roman number 5. So 5 out of 6, that was a clinical diagnosis of Alzheimer's. And this is what the tissue really looked like when you start to look at higher and higher power resolution. Scattered throughout the tissue were the characteristic beta amyloid and tau tangles of Alzheimer's disease. In fact, he had more tau than was typical in other cases we've looked at. And all of these pathology sections show with proper staining the same information. You know, starting with a low-level resolution and working your way up to higher and higher resolutions, it becomes clear that his brain was like Swiss cheese for this kind of pathology, especially in the hippocampus, which is the part of the brain where we encode our memories, and why Alzheimer's patients forget who they are, not just where they left their keys by accident. Okay, here are acknowledgments for the people who helped with Dr. Fishbein and Dr. Vinters, including the residents and so forth. And, but we want to know, what does all this mean now that we understand something about these oldest people? And where can we go next? Well, we want to collect blood samples from these people and their children, because that helps with the analysis of the genome, and you know, perform additional autopsies and develop new methods for treating TTR amyloidosis. And we are in the process of collecting these tissue specimens, and we've now gotten about 10 supercentenarians, collaborating with people at Stanford University and Leroy Hood, who just by coincidence was the director of biology here at Caltech for about 15 years. Yes, but they had a falling out. And so now Hood is the director of a separate Institute for Systems Biology in Seattle and no longer affiliated with the University of Washington. Okay, so here is something about the 260-odd age-related genes that have been identified in humans. It's on a public website. Maybe there's more like 600 genes in the latest incarnation, according to the people who maintain this data. I don't want to teach you biology 101. Molecular biology is a complicated subject matter about how mutations occur, but we want to find the so-called SNPs, the single nucleotide polymorphisms that are relevant to longevity. So we started interviewing these people and drawing blood from them. This is Soledad Mejia in San Diego, and another woman from San Diego, Frederica Moss, who was, by the way, a famous silent film era screenplay writer. I don't know if anybody in the room remembers silent movies, but they were very popular back in the 20s and 30s. We analyzed their blood. I actually got some specimens in liquid nitrogen from 2 women where we did autopsies and saved the pure tissue frozen rather than putting it immediately in formalin, which would have damaged the DNA. And so here I am getting it with Greg Fahey out of a Dewar in liquid nitrogen. I think a couple of people in the audience were with Greg early this morning. We went to Oregon. We got another tissue sample from the 111-year-old in Cresswell, Oregon. Her mother, her daughter and her granddaughter there. And this is what the blood looks like. And we went on a trip to the East Coast. We got blood from Lila Denmark, who is a physician in her early career as a pediatrician who developed the first, I guess, pertussis or whooping cough vaccine. Another lady in Monroe, Georgia, with her son, Sidney Cooper, and she is the second oldest person in the world. In Shreveport, Louisiana, we drew blood from Mississippi Wynn, who is like the 5th or 6th oldest person in the world. And the oldest person currently in the world in Jacksonville, Texas, was Eunice Sanborn. In Europe, we drew blood from Germaine, who spoke only French, but we had translators to help us with that. And one person that we would love to get is the oldest man in the world, Walter Bruning, who lives in Montana. We've been to visit him, But we haven't gotten permission from him yet to draw his blood because right now is the flu season, and he is not seeing any visitors, even from his hometown of Great Falls. Here he is in the 2011 Guinness Book of Records. I drew blood from a local gentleman in the San Fernando Valley who spoke only Farsi because he was born in Tehran. But we have good documentation that he's 113, and we got his blood the other day. There's a bet going on now between myself and Robert Bradbury, who lives in Boston, having different points of view about what we're going to learn. He says, you're not going to learn anything by doing this sequence. And so the bet is scheduled to end on 2014 in 4 more years to see, with 10-to-1 odds, by the way. And anybody in the audience who wishes can join in on this wager. David Kekich has been appointed to be the official custodian for the funds. Somehow we have to put together a committee of 3 people to decide whether, if we publish something, that it's really what we said it was. In a peer-reviewed journal, of course. Now, last couple of slides. 10,000, maybe 100,000 years ago, average life expectancy was 18. Then ancient Egypt. Things have been getting better, and presently it's moving toward the early 80s. But according to what I have tried to explain, there is this invisible fuzzy barrier that will cut you off from living past Jean Calmet, the Calmet limit. But maybe if there's a singularity, according to Ray Kurzweil, we will have an escape velocity. Of course, with nanotechnology, and I'm counting on people in this room to help with that, people at NYU, people at Berkeley, and other research centers around the world, we have to clean out some of the other bad things in our blood vessels that are accumulating. So we need a bridge plan to get us from where we are here today onto the other side at the time of the singularity so that we can take advantage of these new discoveries that the people in this room are going to create for us. And we really would consider it a shame, the people with the gray hair in the room, that we didn't make it until the time of the singularity, and we were the last generation to fail to benefit from these new discoveries. And that's it.